So at Monsanto, we recognize that reliably and sustainably feeding a growing global population is really the most significant challenge of our time. And I'm going to spend a couple of slides right up front just talking to you a little bit about that and setting some context, but spend most of my talk focusing on how we're using the precision phenotyping capabilities of the automated greenhouse to really help us make better, earlier decisions about the materials and products that we advance for development, for final commercialization, but also making those early decisions to cull products from the pipeline that are a waste of time and are not going to work for ultimate product development. So as we all know, ag is really at the center of a lot of global change. 25 to 30 years from now, there are going to be 2 billion more mouths to feed on this planet. And it's also a great time of optimism. More than any other time in human history, a greater percentage of the human population is pulling itself out of poverty into prosperity. And when they do that, they begin to stabilize their diet and begin consuming more protein, which adds pressure on agriculture and the future of agriculture as more soy and corn is being shifted to produce animal feed instead of feeding people. So if we continue with our current dietary practices and waste production practices in the West, we waste up to 40% of the agricultural produce that we generate. If we continue on that trajectory, we'll need to double food supplies by 2050. Even the most optimistic projections of waste reduction and changing diets put us at about 60%. So we need to grow more, but we can't just grow more on existing natural ecosystems. We have to grow more on existing agricultural land. And overlaying all of this is the uncertainty of climate change. We don't know exactly how climate change is going to impact the future of agriculture, but it will impact it. We know at this point that it's likely to shift growing regions to the north. Along with that, we'll probably see a shift in insect, weed, and disease infestations further north. And we're also going to see an increased frequency of severe weather events like drought and heat stress. So we're going to need to develop reliably yielding products in a climate of extreme uncertainty. And we're really on the right track. We've made significant progress if we just look at corn in increasing yield on existing acreage. So if we look at corn yield prior to the advent of the hybrid era, it took a land equivalent of about half of a basketball court to produce a single bushel of corn. With the advent of hybrid breeding in the late 1930s, between 1939 and 1990, we saw an increase in yield of about 4 to 5-fold. And then with the advent of biotechnology in the 90s, we continued to see increased yield, but importantly, we started beginning to see that stability, year-on-year stability. So biotechnology brought a stability to yield that breeding alone wasn't able to do. So we've gone from half a basketball court for a bushel of corn to where we are today, where we can grow a single bushel of corn in a land area equivalent of about the space inside the free throw lane. So that's really significant progress, but we need to do more. We need to reduce that footprint by another 25 to 50% in the 25 to 30 years ahead. So at Monsanto, we recognize that there's really no single magic bullet to doing this. And we partner broadly with academics and industrial collaborators to really identify the biotech products, the breeding germplasm, the biological and chemistry, foliar and seed treatments that will really bring us to the next generation of agricultural products. And overlaying all of this, we're working with advanced data analytics to help us understand how materials are performing in in the field and really help farmers make the best agronomic decisions for the products that we're developing. And that's really where the automated greenhouse comes in terms of identifying things that work that we can advance for commercialization. So the automated greenhouse is really a precision phenotyping system that allows us to, with very high confidence, characterize the things that we're introducing early on into the product development pipeline. So as a complex moving system, what I wanted to do is just share a video of what the automated greenhouse looks like in operation and talk to you a little bit about some of the high-level details and then go on into some of the deeper details through the remainder of my talk. So we have a promotional video that we developed a number of years ago, and all of the plant handling activities within the greenhouse are fully automated. The greenhouse is about 28,000 square foot under glass, separated into 4 greenhouse growing bays. We can grow a variety of plant species in the greenhouse. Each day, the plants come out of the growth bays into the head house where data is collected on them. We track everything through the greenhouse as an individual. So when a seed goes into a pot, that plant is tracked through the greenhouse. And everything is treated as an individual. So water and fertilizer is applied based on the experimental parameters that are defined by the experimental conditions. We have a number of layers of quality that I'll talk to you a little bit about to make sure that we have the highest confidence in the data that we're generating and the results that are coming out of the greenhouse. And really this combination of automation and automated image-based data collection allows us to operate and phenotype crop species at a throughput that has never before been possible. And that video is on YouTube if you are interested in revisiting it. So the automated greenhouse, as you can see, is really an intersection of a number of different technology areas. And Monsanto, as if anybody knows the history of the RTP site, Monsanto acquired the Research Triangle Park site as part of an acquisition of Paradigm Genetics back in 2005. And at that time, what Monsanto acquired was really the assets on the far right of this slide. So we had a high throughput Arabidopsis phenotyping system and had expertise in plant growth and physiology, automated image analysis, and all of the data systems that underlie that that allowed us to do that at the kind of throughput that we needed to do. And when Monsanto acquired us, they pretty quickly said, okay, we want you to take all of that expertise and instead of applying it to a model plant, apply it to the plants that are going to be the commercial products that we care about. And that's really where the automated greenhouse came in terms of the automation and engineering that went into play that allowed us to bring that kind of throughput that we developed for Arabidopsis to crop species. Really at the heart of the automated greenhouse is advanced imaging capabilities that we customize specifically for phenotyping crop species. So we have customized visible and hyperspectral image algorithms for phenotyping crop species. The algorithms and imaging segmentation routines for visible light imaging allow us to very accurately quantify plant growth and development milestones such as biomass gain, plant height, canopy area, as well as key reproductive milestones like flowering and time to senescence, and also flowering duration, flowering number, flowering duration. We also have hyperspectral imaging capabilities. So this allows us to detect wavelength-specific reflectance in the greenhouse. So we can quantify plant pigment content, so things like chlorophyll content, anthocyanin content to get a read on stress response, as well as foliar water content. And then finally, we also measure the amount of water, so we can start bringing some of these metrics together to calculate things like water use efficiency as plants are going through the greenhouse. As I said, everything is tracked through the greenhouse individually, and that's all done through RFID tracking. So every plant in the greenhouse has an RFID chip associated with it, and we characterize every plant individually in the greenhouse. Since everything's coming through the greenhouse on a daily basis, we get very high resolution. Rate data for plants going through running through an experiment. We have very precise control over how we treat the plants, so we can based on the experimental question that's being asked, we can apply a very specific volumetric water content to start looking at things like performance under drought stress. We can also very tightly control nitrogen to look at nitrogen use efficiency, as well as all of the other macronutrients and micronutrients. And then finally, we have very precise control over the timing and duration of the stresses that we impose on plants. So really the scope of questions that you want to ask is limited by your creativity using the automated greenhouse. We have assays for multiple treatment conditions, as I just mentioned, across nitrogen, water. We can titrate and also do dual stresses. We've also got imaging algorithms for multiple species. So the greenhouse was built to evaluate corn, soy, and cotton. But it turns out the imaging algorithms that we've developed are quite robust, and with very little modification can be extended to other plant species as well. And we're also looking at the full spectrum of growth stage. So we can now run corn in the greenhouse from seed to seed if the experiment demanded it. Quality really is at the heart of a system as complex as this, and I can't overstate that enough. The success of everything that we produce in the automated greenhouse relies on a number of layers of quality at every level of operation. The greenhouse is an ISO certified facility, and as an ISO certified facility, we have a number of key performance indicators that ensure operational integrity throughout the operation of the greenhouse. Things like fertigation volume being added, the imaging, the white balance imaging standards, simply the conveyor system in the greenhouse, things like that. And by having that really at the heart of everything that we do, it's really become indoctrinated to the people working at the site that quality oversight is something that we eat, sleep, and breathe for the data that we generate. Assay stability is monitored by running a consistent known through the greenhouse every time we sow plants in the greenhouse. So we monitor a known and make sure that it's within specific experimental parameters throughout the course of a growing year. We validate every metric that we bring into the greenhouse. I'll talk a little bit about that in the next slide. And then finally, we have very tight oversight of experimental design, resulting data QC, and then partnering with customers that are bringing experiments into the greenhouse for the visualization step. So making sure that they're getting the visualization that they need in order to turn the data that we're generating into knowledge that they can use. Metric validation is something that we do for every single metric that we capture in the greenhouse, whether it's a visible light metric like biomass, like I'm showing on this slide, or a flowering metric for reproductive phenotyping, or one of the hyperspectral metrics for chlorophyll content. And this is what metric validation looks like for biomass, for example. What we do is we'll take plants through the greenhouse and calculate their biomass using image-based tools, and we'll also harvest the actual shoot biomass and compare the image-derived metric with the ground truth metric. So what we're looking at here is biomass gain over time for plants, corn plants grown under 3 different treatment conditions that are represented by the colors there. So that's an overview of the automated greenhouse facility. And what I'd like to do now is show you a little bit about what that looks like in practice. And I'm going to use the example of drought, since drought is really the most significant challenge that corn faces year on year. I'm going to show you how we could use the greenhouse to identify drought products in the categories that I talked about a little bit earlier. A drought is something that we, of course, really care about. We underwent one of the most significant droughts of the century in 2012. And what we would like to do at Monsanto is develop products that are going to provide farmers a drought insurance in some of these moderate to severe drought areas, and also provide farmers with survivability insurance for short-term drought in the more extreme and exceptional growing areas that may be of shorter duration. So the first thing I want to talk about is how we could use the greenhouse to identify drought-tolerant germplasm. And this is just an example from an experiment that we ran recently, where we ran 55 different corn hybrids through the automated greenhouse under standard and water stress conditions. And what you can see is that there's three, oops, keep pressing the wrong button there. There's 3 or 4 here that are showing exceptional biomass accumulation under drought conditions, and that would be germplasm that we would want to advance for developing a drought-tolerant product. In contrast, on the other end of the spectrum, you can see 4 or 5 hybrids that are showing increased sensitivity to drought. And again, importantly, those are hybrids that we would want to cull from a drought development, product development pipeline early, so we don't waste a lot of time and resources on them down the road. Similarly, we're using the automated greenhouse to identify biotech products for drought tolerance. And this is just an example of that. So here we're looking at 3 independent events of a gene that's been introduced as a discovery effort for drought tolerance. And all 3 of the events show increased biomass under water deficit conditions, as well as increased water use efficiency. And what we're looking at here are deltas versus the control. So we're seeing a 14 to 15% increased biomass versus the non-transgenic control under water deficit conditions in these 3 events. And finally, we can also use the automated greenhouse to identify seed treatments and foliar applications and chemistry from chemistry and microbial product lines that can improve growth under water deficit conditions. So this is an example in that category. And what we're seeing here are based on a single seed treatment that we're working with under standard conditions. The treated and untreated plants grow essentially identically. Under water deficit conditions, the treated seeds show increased biomass gain at a very early time point and continue to outpace the untreated seeds throughout the duration of the untreated plants throughout the duration of the experiment. So the greenhouse can really be used to identify products that we can then bring together to help farmers make best decisions for the material that they're planting on their acres. And what I hope I've communicated to you is that we can really use this kind of precision phenotyping to very early on in the product development pipeline identify the things that are working well, eliminate the things that are working poorly, and really make early decisions that are going to impact later product development. And with that, I am finished.